Next story. Revolutionary DNA origami vaccines for precision cancer immunotherapy. In a groundbreaking discovery by researchers at the Wyss Institute for Biologically Inspired Engineering at Harvard University, a revolutionary DNA origami platform named DoriVac has been unveiled. This platform utilizes self-assembling nanostructures to deliver precise spacing of adjuvant molecules that stimulate immune cells to produce a robust anti-tumor response. By attaching adjuvant molecules to a square block-shaped nanostructure, researchers found that spacing these molecules 3.5 nanometers apart yielded the most effective activation of antigen-presenting cells, APCs. These activated APCs then induced a desirable profile of T cells, including cytotoxic T cells that kill cancer cells, Th1 polarized T cells that cause beneficial inflammation, and memory T cells that provide long-term immune memory of the tumor. DoriVac vaccines administered to tumor-bearing mice demonstrated enhanced control of tumor growth and significantly increased survival rates compared to control mice. Furthermore, when combined with immune checkpoint inhibitors, a widely used immunotherapy, the effects of DoriVac were amplified, leading to complete regression of melanoma tumors and prevention of regrowth upon re-exposure to tumor cells. This innovative approach not only shows promising results in preclinical mouse models but also has the potential for personalized cancer vaccines tailored to individual patients. By loading antigens onto prefabricated DNA origami structures, researchers aim to develop highly effective cancer vaccines that can be used in conjunction with existing immunotherapies for improved clinical outcomes. The implications of this research are far-reaching, offering a new frontier in cancer immunotherapy that leverages nanotechnology to precisely modulate immune responses against tumors. The development of the DoriVac platform represents a significant advancement in the field of precision medicine, bringing us closer to the realization of personalized and effective cancer treatments for patients worldwide. Source: Eureka Alert. Next story. Unlocking the Arctic nightlife. Little Auk's nocturnal chorus revealed through acoustic recordings. In the heart of the Arctic wilderness, where endless daylight reigns supreme during the summer months, a vibrant colony of little auks has been caught in the act. Researchers from Hokkaido University and Aarhus University have embarked on a mesmerizing journey into the nocturnal world of these seabirds, uncovering surprising revelations about their hidden rhythms under the midnight sun. With cutting-edge acoustic and imaging technologies at their disposal, the scientific team set out to decode the enigmatic daily routines of the little auks in northwest Greenland. What they discovered was a symphony of sound as the birds engaged in a nocturnal chorus unlike anything witnessed by those accustomed to the rhythms of mid-latitudes. Led by the visionary associate professor Evgeny A. Podolsky, the study, recently published in Communications Biology, showcases how these little auks adapt to life in the Arctic, synchronizing their vocalizations with the behavioral patterns of attendance, feeding, and fledging. Their vocal activity peaks at night, a stark contrast to the dawn choruses familiar to many. In a region where over 60 million birds convene each summer for breeding and foraging, the researchers emphasize the critical role of passive acoustic monitoring in unraveling the mysteries of avian behavior in remote and challenging terrains. Dr. Anders Mosbeach, a co-author from Aarhus University, underscores the importance of understanding these dynamics for the conservation and management of Arctic ecosystems in the face of rapid environmental changes. Monica Ogawa, a PhD candidate at Hokkaido University and co-author of the study, highlights the significance of combining audio data with other monitoring techniques to bolster conservation efforts for seabird populations. By tapping into the power of interdisciplinary collaborations and engaging local communities, the research team aims to pave the way for sustainable practices that safeguard these vital avian species. As the researchers delve deeper into the acoustic ecology of Arctic seabirds, their mission transcends mere scientific curiosity. They seek to unravel the intricate web of interactions that sustain life in one of the planet's most extreme environments, shedding light on the complex dance of nature in the far reaches of the north. Through their groundbreaking work, the team not only offers a rare glimpse into the nocturnal world of little auks but also champions the use of advanced technologies to study wildlife behavior in challenging landscapes. Their efforts serve as a testament to the power of innovation and collaboration in unraveling the secrets of the natural world, promising a brighter future for conservation and ecological understanding in the Arctic realm. In the realm of the Arctic, where the sun never sets and the wildlife thrives in the face of adversity, the little auks stand as sentinels of a changing world, their nocturnal symphony echoing across the vast expanse of the north. Source: Eureka Alert. Next story. Uncovering abnormal brain patterns in kids with language development issues, a breakthrough study. In a groundbreaking study published by Georgetown University Medical Center, researchers have identified abnormalities in a specific part of the brain associated with movement in children with developmental language impairments. This discovery, featured in the prestigious journal Nature Human Behavior, has the potential to revolutionize the diagnosis and treatment of language difficulties in children. The study focused on developmental language disorder, a common condition that affects various aspects of language development. Through an extensive analysis of 22 articles investigating brain structures in individuals with the disorder, researchers pinpointed abnormalities in the anterior neostriatum within the basal ganglia, a crucial brain region responsible for motor control and learning. Lead author Dr. Michael T. Ullman, a renowned neuroscience professor, highlighted the significance of these findings in raising awareness about developmental language difficulties. The research suggests that drugs targeting dopamine receptors, known to improve movement impairments related to basal ganglia dysfunction, could potentially benefit children with language impairments. Furthermore, interventions promoting compensation by unaffected brain regions could offer additional therapeutic avenues. The identification of basal ganglia abnormalities as potential early biomarkers for developmental language problems opens up possibilities for early intervention and therapy, potentially transforming outcomes for affected children. The study emphasizes the urgent need for continued research into the neurobiology of developmental language disorders, particularly focusing on the role of the basal ganglia. Collaborating with researchers from Deakin University and the University of Minnesota Medical Center, the study was supported by grants from the NIH and NSF, as well as funding from the Mabel H. Flory Trust. Georgetown University Medical Center's dedication to excellence in education, cutting-edge research, and patient care underscores its commitment to advancing human health. Through a holistic approach rooted in social justice and the Jesuit principle of cura personalis, GUMC stands at the forefront of medical and scientific innovation. As the scientific community delves deeper into the neural mechanisms underlying developmental language disorders, the potential for transformative interventions and improved outcomes for affected children looms on the horizon. Source: Eureka Alert. Next story. Revolutionizing ultraviolet spectroscopy. Physicists make groundbreaking progress in precision and accuracy at minimal light levels. In a groundbreaking achievement, physicists have shattered previous limitations in ultraviolet spectroscopy, pushing the boundaries of precision and accuracy to unprecedented levels at extremely low light intensities. This milestone, published in the renowned journal Nature, marks a significant leap forward in the realm of scientific discovery. Utilizing an ultraviolet photon-counting dual-comb spectrometer, researchers at Max Planck Gesellschaft have harnessed the power of two ultraviolet frequency combs with slightly different pulse repetition frequencies. Operating at remarkably low light levels generated through nonlinear frequency conversion of near-infrared combs, this innovative approach has revolutionized the field of spectroscopy. The ultraviolet combs, each carrying a faint pulse of light, pass through a sample before being combined and detected by a photon-counting detector. What sets this advancement apart is its ability to analyze samples at power levels over 1,000,000 times weaker than what was traditionally used. Despite the minuscule amount of light, the statistical properties of the detected photons contain a wealth of information about the sample's optical spectrum, potentially revealing intricate details that were previously inaccessible. This pioneering technique opens up a realm of possibilities for researchers across various disciplines. By harnessing the statistical data embedded in these faint photons, scientists can delve deeper into the molecular composition of substances, explore complex optical properties, and unlock new avenues for understanding fundamental phenomena at a microscopic level. The implications of this breakthrough extend far and wide, promising to revolutionize fields such as materials science, chemistry, biology, and beyond. With this newfound capability to probe samples at such extraordinary sensitivity, the scientific community is poised to make significant strides in unraveling mysteries that have long eluded detection. As we stand on the cusp of a new era in ultraviolet spectroscopy, the fusion of cutting-edge technology with boundless curiosity propels us towards a future where the invisible becomes visible, and the once imperceptible details of our universe are brought to light in dazzling clarity. Source: Eureka Alert. Next story. Gut fungal changes, key biomarkers for liver disease progression. In a groundbreaking study published by Science China Life Sciences, the research team led by Langquan Li from Zhejiang University delved into the intricate world of gut fungal alterations and their potential as biomarkers for primary liver disease progression. The team's findings shed light on the correlation between longitudinal changes in the gut microbiome and the development of liver diseases, particularly hepatocellular carcinoma, HCC. Utilizing mouse models of hepatocarcinogenesis induced by DEN and carbon tetrachloride, the researchers observed a fascinating pattern of fungal abundance shifting from Chytridia mycota to Ascomycota during the progression of liver disease, with Kazakhstania pintolopeci emerging as a dominant species in HCC cases. Subsequent analysis of fecal samples from liver disease patients corroborated these findings, emphasizing the disrupted Ascomycota in HCC and the increased abundance of the Candida genus in HCC patients compared to those with liver cirrhosis. Further investigations explored the effects of specific fungi, such as C. albicans and S. cerevisiae, on the progression of liver diseases. Administering these fungi to mice revealed varying impacts on tumor growth and disease advancement, indicating the potential of gut microbiota not only as diagnostic biomarkers but also as viable targets for preventive and therapeutic interventions. This study marks a significant stride in understanding the role of gut fungi in liver disease progression, offering insights that could revolutionize clinical diagnosis and treatment strategies. The identification of specific fungal biomarkers for HCC presents a promising avenue for early detection and intervention, potentially transforming the landscape of liver disease management. As the scientific community continues to unravel the complexities of the gut microbiome and its impact on human health, studies like this serve as a beacon of hope for individuals at risk of liver diseases, paving the way for more personalized and effective approaches to disease prevention and treatment. Source: Uric Alert. Next story. Exciting news. Zoonoses Journal joins Brazilian Society for Virology. In a groundbreaking announcement on March 15, 2024, Zoonoses, an open-access journal, has proudly declared its affiliation with the prestigious Brazilian Society for Virology, SBV. The move marks a significant milestone in the field of virology, as Zoonoses becomes an integral part of the SBV network, aiming to foster scientific collaboration and raise awareness about the critical health risks associated with zoonotic diseases. With a mission to share scientific insights, encourage international partnerships, and educate the public on zoonoses, Zoonoses has waived article processing charges during its initial launch phase. Furthermore, SBV members will enjoy a 20% discount on article processing charges once they are introduced. Catering to a diverse audience comprising research scientists, physicians, veterinarians, and public health experts, Zoonoses invites submissions for cutting-edge research in zoonotic diseases. The journal is now accepting articles online, offering an open platform for professionals to contribute to the field's advancement. To facilitate easy access and engagement, Zoonoses can be found on Science Open, providing a hub for researchers to explore and interact with the latest publications. Authors can conveniently submit their work using Scholar One, without facing any submission or article processing fees. For those eager to stay updated on Zoonoses' latest developments, the journal can be followed on Twitter, Facebook, and LinkedIn, allowing for real-time updates and community interaction. The journal's editorial board, comprising leading experts in the field, ensures rigorous peer review and high-quality publications. As an open-access journal, Zoonoses remains committed to disseminating knowledge and fostering collaboration across borders. By embracing transparency and accessibility, the journal aims to drive innovation and understanding in zoonotic diseases, ultimately benefiting global health outcomes. For more information on Zoonoses and its affiliation with the Brazilian Society for Virology, visit the journal's official website and explore the wealth of resources available to the scientific community. Stay tuned for the latest breakthroughs and insights as Zoonoses continues its journey towards advancing virology research and public health awareness. Source: Uric Alert. Next story. Stratospheric discovery. Weak polar vortex improves forecasting in Northern Europe. In a thrilling tale of atmospheric dynamics and meteorological marvels, researchers at Ludwig Maximilians Universität München have unveiled a groundbreaking discovery that is set to revolutionize long-range weather forecasting in Northern Europe. Delving into the stratosphere, a layer of the atmosphere above our heads, these astute scientists have unearthed a hidden connection between the polar vortex and weather predictability. When this swirling band of strong eastward flow in the stratosphere weakens or breaks down, a cascade of effects is set in motion. The North Atlantic jet stream takes a southerly detour, paving the way for cold snaps over Eurasia, a phenomenon that occurs roughly every other winter. As the polar vortex currently undergoes a weakening episode, the stage is set for a transformation in long-range weather forecasts. Researchers have observed that during periods of a feeble polar vortex, uncertainty in predicting weather conditions over northern Europe diminishes significantly. Ensembles of forecasts now show a remarkable 25% reduction in the range of potential weather outcomes, offering a more focused glimpse into the future climate landscape. Jonas Spaeth, a doctoral student at LMU, underscores the impact of the North Atlantic jet stream's southern shift on this newfound predictability. The altered tracks of winter storms, the primary source of forecast uncertainties during this season, lead to decreased storm activity and subsequent forecast precision over northern Europe. However, as the pendulum swings, forecast uncertainty gains ground over southern Europe, showcasing the delicate dance of atmospheric dynamics. In a recent study published in Communications Earth and Environment, LMU meteorologists shed light on the intricate dance between the stratosphere and surface weather, highlighting the tangible benefits of unraveling these remote atmospheric connections. With a deeper understanding of these phenomena, the practical utility of long-range forecasts receives a significant boost, enabling various sectors such as agriculture, energy, and public health to brace for impending weather fluctuations. As the world of meteorology embraces this groundbreaking revelation, the future of weather forecasting in northern Europe appears brighter and more predictable than ever before. With the subtle nuances of the stratosphere painting a clearer picture of the atmospheric symphony, the chaos of weather prediction gives way to a harmonious melody of precision and foresight. In a world where every degree of accuracy counts, the realm of meteorology stands on the cusp of a transformative era, guided by the keen insights and tireless efforts of the researchers at LMU. Prepare to bid adieu to the vagaries of weather prediction and usher in a new age of meteorological enlightenment, where the secrets of the stratosphere unveil a roadmap to weather forecasting precision. Source: Uric Alert. Next story. Inactive smokers harbor rich microbial communities, study finds, in the mysterious depths of the ocean floor, where total darkness reigns and pressures are extreme. An international team of researchers from MAREM, Center for Marine Environmental Sciences at the University of Bremen, is unraveling the enigma of microbial life in seemingly uninhabitable areas. Their investigation focuses on inactive hydrothermal vents, once thriving hubs of biodiversity fueled by energy-rich fluids. These vents, known as black smokers, form chimneys where mineral-rich waters meet the cold sea, creating a unique ecosystem based on chemosynthesis. As the hot fluid source dwindles, larger organisms migrate, leaving behind resilient microbial communities. Dr. Florence Schubotz, leading the research, reveals the surprising discovery that even dormant smokers teem with microbial activity, playing a crucial role in organic carbon production on the ocean floor. This groundbreaking study sheds light on the intricate carbon cycle of the deep sea, highlighting the vital importance of these ecosystems. Sampling expeditions conducted in the eastern Pacific, where the first hydrothermal vents were found, unveil the density of microbial colonization in both active and inactive smokers. The team's meticulous work, aided by manned submersibles like Alvin, aims to understand the metabolic changes in these deep-sea organisms and their impact on biogeochemical cycles. These findings are crucial for assessing the potential influence of inactive chimneys on the deep-sea ecosystem and guiding decisions regarding deep-sea mining activities. The research not only deepens our understanding of the ocean's hidden realms but also underscores the necessity of safeguarding these delicate ecosystems. MARUM's commitment to uncovering scientific truths about our oceans aligns with the United Nations Sustainability Goals, emphasizing the importance of preserving marine environments for future generations. By sharing their findings with the public, MARUM aims to foster awareness and dialogue on marine conservation and sustainable research practices. As we delve deeper into the mysteries of the deep sea, each discovery brings us closer to unraveling the intricate tapestry of life that thrives in these extreme environments. The microbial communities that persist in inactive smokers challenge our perceptions of habitability and resilience, showcasing the remarkable adaptability of life on Earth. Through collaborative efforts and cutting-edge research, we inch closer to unlocking the secrets of the ocean floor and understanding our planet's interconnected ecosystems. Source: Uric Alert. Next story. Revolutionary flexible sensors. zero Poisson's ratio for advanced biaxial detection. In a groundbreaking development in the field of flexible sensors, a Chinese team of scientists has introduced a novel approach to revolutionize sensor technology. Published in the prestigious National Science Review, the research by Professor Hao Wu of Huazhong University of Science and Technology presents a game-changing strategy: metamaterial membranes with zero Poisson's ratio. These innovative membranes can independently detect biaxial stimuli, marking a significant advancement in flexible sensor capabilities. The traditional challenge with flexible sensors has been their limited ability to accurately measure complex deformations resulting from forces or strains acting on multiple axes. The key hindrance has been the Poisson's effect, where materials exhibit a change in width when subjected to longitudinal strain. By introducing zero Poisson's ratio, ZPR, materials that maintain a constant transverse width under strain, the research team has overcome this limitation, enabling the independent perception of biaxial stimuli. Creating ZPR elastomer membranes posed a formidable challenge due to the inherent properties of elastomers. To achieve this feat, Professor Wu and Dr. Xin Huang devised a unique approach by combining positive Poisson's ratio, PPR, structures with negative Poisson's ratio, NPR, structures to create a hybrid membrane with zero Poisson's ratio. Through meticulous experimentation and finite element analysis, the team successfully designed ZPR membranes with a Poisson's ratio close to zero, showcasing the effectiveness of their approach. The flexible sensors based on these ZPR membranes exhibit remarkable capabilities. They can accurately detect uniaxial stimuli and independently perceive biaxial stimuli, a feat previously unattainable with conventional sensors. When subjected to uniaxial stretching, the sensors demonstrate a linear increase in electric resistance along the stretching direction, while resistance perpendicular to the stretch remains relatively constant. This selective response allows for the precise detection of stimuli along specific axes, enabling sophisticated applications in robotics and motion tracking. The ZPR flexible sensors offer a wide array of applications, including robotic manipulation, healthcare, human-machine interfaces, and tactile sensing in soft robotics. By accurately measuring contact forces, detecting motion status, and monitoring deformations, these sensors open up new possibilities for enhancing robotic capabilities and human-computer interactions. Their ability to detect subtle changes in force, strain, and motion direction makes them invaluable tools for a variety of industries and domains. Professor Wu envisions a future where ZPR sensors play a pivotal role in advancing technology and revolutionizing how we interact with machines and devices. With their exotic sensing capabilities and versatility, ZPR flexible sensors are poised to drive innovation and enable new frontiers in sensor technology. The research published in National Science Review heralds a new era of flexible sensors, paving the way for transformative applications across industries and disciplines. The fusion of cutting-edge materials science with sensor technology has unlocked endless possibilities, propelling us into a future where precision, flexibility, and independence in sensor capabilities reign supreme. Source: Uric Alert. Next story. Revolutionizing CAR T-cell production with virus-like nanovesicles. In a groundbreaking development, a group of researchers led by Professor Zheng Wang and Professor Kong-Fei Zhu from the School of Biomedical Sciences and Engineering at South China University of Technology has devised an innovative technique for producing CAR T-cells in vivo. The key lies in virus-mimetic fusogenic nanovesicles, FuNV, loaded with anti-CD19, alpha-CD19, chimeric antigen receptor, CAR, molecules, which seamlessly fuse with T-cells, catalyzing the conversion into potent CAR T-cells within the body. CAR T-cell therapy has demonstrated remarkable success in battling B-cell lymphoma and leukemia. However, the intricate and costly process of manufacturing CAR T-cells has impeded its widespread adoption. This new approach eliminates the need to harvest T-cells from patients by directly transferring CAR molecules from FuNVs to T-cells. By integrating the CAR protein onto T-cells through FuNVs, the risk of complications like cytokine release syndrome and tumorigenicity associated with viral gene integration is mitigated. The researchers ingeniously engineered T-cell fusogens by combining an anti-CD3 fragment with fusogens from reoviruses or measles viruses. This modification enabled FuNVs derived from these cells to efficiently induce fusion with T-cells, both in lab settings and in living organisms. By expressing the fusogen and alpha-CD19 CAR protein in T cells, they produced FunVs loaded with CARs, FunVCAR, that successfully generated CAR T cells in vitro and in vivo. Moreover, intravenous administration of FunVCAR significantly impeded the growth of B-cell lymphoma. Extensive toxicity evaluations revealed that FunVCAR did not induce adverse effects on blood composition, body weight, or inflammatory cytokine levels, unlike traditional CAR T-cell treatments. The transient activation of CAR T-cells produced by FunVCAR resulted in controlled cytokine release, distinct from the sustained inflammation observed with conventional methods. This pioneering study heralds a new era in CAR T-cell production, offering a streamlined and safer alternative for patients. While immensely promising, this approach may not be suitable for individuals with compromised T-cell functionality. The fusion of cutting-edge nanotechnology with immunotherapy opens doors to more accessible and effective CAR T-cell therapies, bringing us one step closer to revolutionizing cancer treatment. Source: Eureka Alert. Next story. Aging monkeys. Study shows decrease in social connections over time. In the heart of Thailand's lush Phu Kieo Wildlife Sanctuary, a tale of social evolution unfolds as scientists delve into the intricate world of aging Assamese macaques. At the German Primate Center, researchers uncover a fascinating discovery. Just like humans, these primates too experience a shift in their social dynamics as they mature. The story begins with the realization that as individuals age, their focus hones in on a select few important relationships. This phenomenon of social restructuring towards close kin and intimate companions is not limited to our human existence but is deeply ingrained in our evolutionary past. Through their extensive field studies spanning 8 years, the team of scientists observed a compelling trend among the female Assamese macaques, a gradual retreat from extensive social interactions as they aged. Contrary to the notion that aging equates to a solitary existence, the study revealed that older female macaques maintained proximity to their group members but opted for fewer social engagements. The intricate web of social connections these primates weave mirrors the complexities of human relationships, shedding light on the biological underpinnings of social aging. As the lead researcher, Baptiste Sadiki unveils the secrets of the macaque's social evolution, he uncovers a poignant truth. The essence of selectivity in social bonds remains a constant, yet the frequency of interactions dwindles with age. This intriguing dichotomy challenges traditional beliefs and beckons us to delve deeper into the mysteries of social aging. In a world where social bonds are the bedrock of well-being, the study offers a unique perspective on how individuals navigate the intricate tapestry of relationships with advancing age. Through meticulous observations and unraveling of behavioral patterns, the researchers decode the age-old enigma of social dynamics in the primate kingdom. As the sun sets over the verdant expanse of the Phu Kieo Wildlife Sanctuary, the echoes of this groundbreaking study resonate far beyond the confines of the forest, touching upon the very essence of what it means to forge lasting connections in the twilight years of life. With each revelation comes a deeper understanding of the evolutionary threads that bind us to our primate cousins, illuminating the path towards unraveling the mysteries of social aging in both human and non-human societies. As we embark on this illuminating journey of discovery, the echoes of the macaque's whispers remind us of the timeless bonds that transcend age and species, weaving a tapestry of interconnectedness that unites us all in the grand symphony of life. Source: Eureka Alert. Next story. Achille Mbembe wins 2024 Holberg Prize, a recognition of excellence. In a momentous announcement shaking the academic world to its core, political theorist Achille Mbembe has been crowned as the prestigious 2024 Holberg Prize Laureate. The Cameroonian scholar, a beacon of intellect and innovation, has been long recognized for his groundbreaking contributions to African history, post-colonial studies, and the intricate intersections of humanities and social sciences. Having spent 4 decades delving into the complexities of colonialism, decolonization, and the evolving global landscape, Mbembe's work stands as a testament to his unparalleled ability to intertwine past narratives with pressing contemporary issues. From migration regimes to climate change, his vision transcends boundaries, pioneering new paths for understanding and reimagining our world. Mbembe's influential oeuvre, translated into 17 languages, captivates readers worldwide. His seminal works such as On the Post-Colony, Necropolitics, and The Earthly Community have sparked transformative dialogues on power structures, life, and mortality. Through his concepts of necropolitics and the universal right to breathe, Mbembe challenges conventional wisdom, reshaping our perceptions of identity, dignity, and shared humanity in a racially charged world. As a critical theorist, Mbembe's quest for a planetary consciousness resonates profoundly. His relentless pursuit of open futures beyond historical shackles of race and segregation ignites a flame of hope for a world built on repair, care, and universal solidarity. Reflecting on his life's work, he poses poignant questions, urging us to reimagine our existence, our connections, and our collective responsibility to nurture a sustainable and inclusive future. Mbembe's accolade as the 2024 Holberg Prize Laureate signifies a pivotal moment in academia, honoring his exceptional scholarship and intellectual prowess. His unwavering dedication to historical truth and South-North dialogue embodies a universalist ethos, inspiring generations of scholars to challenge conventions, embrace diversity, and envision a world where knowledge transcends borders. As we celebrate Professor Achille Mbembe's remarkable achievement, we recognize him not only as a scholar and philosopher but as a visionary whose legacy will continue to shape the intellectual landscape for years to come. His narrative is a testament to the power of knowledge, the beauty of diversity, and the boundless potential of human thought. The story of Achille Mbembe, the 2024 Holberg Prize Laureate, is a tale of resilience, innovation, and a commitment to a better tomorrow, a narrative that will echo through the corridors of academia for generations to come. Source: Eureka Alert.